All right, everybody, let's get back. So this is what's interesting about this particular unit. It, I'm the PowerPoint just keep giving me an idea of what I want to cover. The reality is when I even for the exam going forward, it's typically how I would describe it, kind of like a read your PowerPoint kind of study guide thing. I mean, it's really I'm taking most of the information off the PowerPoint, so that I would describe it to you. And so I'll be a few little nuggets that I'll throw in there that are important, but that's really where the bulk of the information is coming from. It must be something I said. I've never seen so few people in this class. You know, it's okay. Sydney's in trouble. We'll just Brooke, I can deal with it. All right. So a few people online. At least you're here. Okay. This is all about how infections occur. This is about how they come in, how they go out, what they do on the inside, how they kind of do their magic, generically speaking. So this is that guide about what happens, and then at the end of it, it's epidemiological aspects of it, how they spread, the risks and gains vis-a-vis how they spread, all of those kinds of things. There's a lot of very useful and particularly useful information from folks who are going to be dealing with whatever your role might be in the health, assuming you're in the healthcare tree, whatever your role is. It could be nursing, it could be any other clinically related profession. There's a lot of public health implications that are part of it, and that became more relevant. We sort of went away from aspects of public health, and then when COVID came around, it became more of an interest. I mean, there's some interesting sort of offshoots that are associated with this, and a fair amount of definitions as well. So moving on, how it enters, portal of entry, how it gets in. Okay, and most of it now they can come from somewhere already in your body. That's not that unusual. We did throat, and I did throat cultures, and you did throat cultures in your lab. I mean, that's a potentially common source from nose and throat. I mean, a lot of those are endogenous. Exogenous is from somewhere outside. So those are pretty straightforward terms. And this is, even though and there's some things that really bear commenting on. Okay, most of these agents, most of the organisms, or viruses for that matter, so pathogens is probably a better generic term, specific portal of entry that's favorable for them to grow in. Okay, this is true, but requires clarification. So if it enters the quote wrong way, it will not be infectious. Okay. Look, we get the flu respiratorily. If it gets in, the lining cells is what it adheres to inside your respiratory system, particularly your nose. But if the virus contacts the skin, no infection occurs. That's true, but it doesn't limit the fact that once it's on your skin, as I keep saying to you time and time again, we touch our face all the time. It's pretty easy. Every time we touch our face, now it's like somebody with their hand over there, like you. Now, Zoe, the same thing. Okay, everybody. I just looked and see, and half the students had their hands in close proximity to their faces. It's not a knock. Okay, we all do that. But the reality now, even though that technically is the wrong portal, it's just the nature of the way our behavior is, the way we do things with, sort of automatically without necessarily thinking about them. Unless, oh my God, I've got to be careful about that because of something I might have come in contact with. So it's a little bit different, but yeah, it's true because it doesn't have the receptors. It doesn't mean it doesn't play a role in facilitating its spread. Maybe that's what I want to say. And so we look at things like that, and they'll get to it, but I think it's better to mention it here. We have things that we talk about things that are direct. So when you're within a classroom like this, you're in a fairly close proximity to people. And so if someone happens to be sitting next to you and all of a sudden they start coughing and sneezing out of control, why would you don't want them to come to class if they're sick? If you're within roughly 3 feet on either side, and they start to cough or sneeze, you're going to inhale some of those particles. It's just harm. That's what we call a direct contact. You're in such close proximity. When we talk about something being indirect, Typically, it means if you contact something that a fancy word that you should become familiar with is this, fomite. Fomite, F-O-M-I-T-E, is a generic term for an inanimate object. So it happens differently when they're sitting next to you, when they're sitting here but not next to you. Let's say there's someone in the previous class and started to cough or sneeze. And that material, whatever it is, was temporarily in the air, and why we call them droplets. They drop down, let's say, the tabletop of the chair, assuming that they're still viable, that they still can hurt you. I mean, you just put your hand on it, and I guess you touch your face, and the next thing you know, here you go. That's an indirect contact. So direct means person to person. I mean, obviously, with sexually transmitted, if you think of gonorrhea, okay, or chlamydia, that's going to require direct contact or force. It doesn't exclude completely indirect, but indirect is fairly rare in those cases for a variety of reasons. So, do you understand what I'm saying? You have to understand in a greater context of understanding how diseases spread, there's a lot of what I would call nuances, things that you have to be wary about. So, an agent can be entered by more than one portal, okay? So, tuberculosis you can get from a variety of ways. You can get it even through the gastrointestinal tract. That's unusual. Tuberculosis is an oddball, particularly respiratorily. I've commented on it before, but I really want to amplify the point. It's that COVID, that flu, that RSV, when you cough or sneeze, it's a droplet. It stays in the air temporarily and drops, and drop. There are a couple of things that are we call airborne. They stay in the air. They adhere to tiny fragments of material, microscopic pieces of material that somehow facilitate them staying in the air. Dust particles. That's it. Tuberculosis is one. Another one we touched on in viruses is called Hanta, H-A-N-T-A, and that's a big player in the desert Southwest because you might recall it comes from rodent droppings. Those are airborne. That's why, and there's things in the desert Southwest because sort of like the dust storms that they get, and littered with fungi. That's something called valley fever. And we'll touch on a respiratory Hanta, which we'll look at in respiratory again, tuberculosis and respiratory. Things that are airborne. That's why when you have a TB patient in the hospital, it's, oh my goodness, that's that room where they're going to have the exhaust fan going on, pulling the air out because you don't want to get into the rest of the hospital, the rest of the facility. Why you have to have certain kinds of personal protective equipment. You're going to have those N95 masks on, most likely. I don't know whatever it is today. There's going to be a specific set of precautions for something who's got active tuberculosis. So there's, that's a significant player. Strep and staphylococcus can enter through the skin, urogenitally, respiratorily. Staph can even cause problems with food. The most common kind of food poisoning we get from dairy, staphylococcus. So I mean, they're bandits of the highest order. There's a lot that goes on. So when I talk about from a test perspective, portal of entry, it's the usual portal of entry, the typical portal of entry. And that's the way I would define it. So there's a question, that's the frame of the choice that you would have. So voila. So skin, staph, strep, Clostridium, and strep pyogenes is, again, that's a strep throat strep. I mean, it can be on the skin, it can be respiratory. There's a lot of areas, as you will begin to see. And you can see them. Typical herpes, cold sore. Okay. Lots of these things. Insect bites classify as that. GI is generally from contamination in the food or fluids or via fomite, something that you come in contact with that has it on there. I remember I told you a little bit about that when we had the big problem with hepatitis A at Chi-Chi's in this area, in Beaver Falls, 20 plus years ago. Effectively, the contamination came from being on raw vegetables, in this case being on raw green onions, spring onions, scallions, whatever you want to call them. But that's a fomite, okay? In that case, so it's an inanimate object, it's a plant. In that regard, I realize plants are living, but consider it that way as acting as a fomite. Inhalation, a gazillion things, urogenitally, a lot. And you're going to see these are the areas that I typically cover when we go into specific diseases, which is we start right after this chapter. We go right into respiratory after this. Respiratory, skin, urogenital, and I try to get the GI in and get some wound information as well. A lot of it has to do with this. This is in the context of virulence factors and why things are virulent or nasty. And infectious dose might be number one on the hit parade. Infectious dose, either how little or how much it takes to make you sick. You or generically anyone. The minimum number of microbes necessary to cause an infection proceeds. When you read about it in the literature, they'll talk about things like ID 50. That means the infectious dose at the 50th percentile. You know, what it would take for half of a population to get the dose. Smaller the infectious dose, the greater the virulence. I don't know exactly what Q fever is, but if it's a single cell, I don't want it. But take a look at this. Tuberculosis. Very small infectious dose. What's interesting, we have a lot of mechanisms to prevent that. So it shouldn't make you frightened every time you walk into a room, assuming you have the proper personal protective equipment. But tuberculosis is one of those things where it's more of sustained contact, where even though the infectious dose is low, for one to really get through and facilitate that infection is harder than you think. You might recall me telling you about that student who had, we were talking about her, she was taking care of an aunt at home who had the disease, and they didn't know it. But the fact that she was the caregiver, it caused her not to even have the active disease, but at that point they were treating her for being exposed. That were there. Giardiasis, that's gastrointestinal. That's what my kids had from contaminated water. It didn't take a lot. From well water, when they were staying with the gal who was my office manager, when my first wife and I were away. And that's the one, that coccidiomycosis, when I was telling you about the desert Southwest. That's something that causes something previously known as San Joaquin Valley fever. Now we just call it valley fever. If you live in Arizona, if you live in New Mexico, if you live in the lower or desert areas of southern and California and below, heading into that desert area, Palm Springs, going to San Diego, that area, all there. Okay, that's big problems. That gets in the air. It's very interesting historically. Coccidio, we'll cover that in respiratory diseases. We think that may have been the source for a very advanced Native American race you may or may not be familiar with, who were an established culture 1,000 years ago called the Anastasians. And it's almost like they disappeared overnight. And speaking in large terms, it wasn't like, oh, but they think that they had like a massive dust cloud of coccidio, and within a matter of, let's say less than a year, this culture disappeared. That's why a lot of the buildings, when they excavated, because they buried over time. I think they were mostly in Utah. So it's like those four corner states. I love the history about this. It's fascinating. Gonorrhea, 1,000 cells is a relatively small dose. I cannot tell you how many cases of gonorrhea. It was absolutely rampant in the 70s when I started doing, working in an emergency room. Oh my goodness. I mean, understand, people, until HIV and AIDS became a diagnosis, people did not use condoms. They were only used vis-a-vis pregnancy, if somebody was on the pill or had an IUD or some other means. Imagine how many cases we had. We had STDs. It was probably the most common diagnosis that we had in our emergency room. Yes, ma'am.
So what makes like for tuberculosis 10 cells versus the 1,000 cells, what's different about the organism that makes it more infectious or not?
Once it gets colonized, once it more readily establishes itself, would be the way to describe it. It doesn't, your defense, your body has defenses, but with tuberculosis, because of the nature of it, the defenses can handle only a very tiny amount of tuberculosis. Same thing with gonorrhea. 1,000 cells it's a teeny tiny amount. What you see on your culture plates for bacteria, colonies of a million, and that little tiny drop is in your culture plate. So your immune system can handle a certain amount of those. And it's just, and I say it again, it's a numbers game. I don't know if that explains it, but that's why.
Yeah, is there like differences in the cell or what they produce?
Oh, yeah, yes. For most of them, it has to do with tuberculosis is interesting because it goes inside. Okay. Very, 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 very rare. Once it's inside, it actually multiplies in a white blood cell. So if there's a white blood cell, if the macrophage can't kill it, okay, then it begins to multiply in it. Gonorrhea it attaches to everything. That's why it's so if it isn't gobbled up by macrophages quickly enough, it just adheres to almost anything it comes in contact with. So its virulence factor are these little fimbriae, those tiny things that adhere. I mean, there was a bizarre case. We have a small but select group, and I know that you're modern thinking people. I was hearing a lecture from a rheumatologist. He was talking about how unimaginably quick gonorrhea could spread. He's a rheumatologist, and gonorrhea, if we see an infected joint in an adult, the diagnosis is gonorrhea until proven otherwise. Because it gets into your bloodstream, it goes everywhere in your bloodstream, and it adheres, for whatever reason, it adheres to the interior of joints when it gets there. And he worked in San Francisco. He was from there. And again, it had a fairly large, not to be, again, a fairly large per capita population of gay men. And they were known to have interesting ways of engaging one another. And he found a case of gonorrhea inside the ear. As far as I'm going. Yeah, and I'm a whatever floats your boat kind of a guy. As my daughter would say. Don't even start. As far as I'm going. So I remember telling that to my daughter, and she goes, "What?" Yes, dear. That gives you an idea of gonorrhea. It adheres, and that's trying to be it's a very serious disease. I don't mean to joke about, but I'm trying to point out to you how extraordinarily rapidly it can adhere to things and a variety of tissues. You'll see when we get into a sexually transmitted. Typhoid fever. Okay, now we're getting a little. This was typhoid Mary. I think 10,000 cells isn't a lot. That's a salmonella infection. But cholera, look, 1 million cells. Was that 1,000 here, millions here? Was that 1 billion cells? Yeah, it is. That's a lot of cells. Yeah, cholera is deadly, but it gets into the water supply. And that's not a lot when you're talking about parts per billion and parts per trillion when we look at water supply. That's an immense amount. So again, look at a culture plate. There's nothing to have 1 billion cells. You get a culture plate that's filled with growth, you got 1 billion cells. Easy. Gives you an idea. Here's the adhesion. Microbes gain a stable foothold, and then once they adhere, they can begin to colonize and reproduce effectively. That's where you're talking about with what gonorrhea does. Dependent on specific binding molecule, okay, it's only confined to certain cells. That's the limitation that we're talking about, where respiratory, couldn't necessarily adhere to a skin. My point is the fact that it's on your skin, it's really a hop, skip, and a jump away from respiratory. So firm attachment is almost always a prerequisite for causing disease, because we have so many things. We have mucus, we have saliva, we have urine, we have perspiration. All of those things are fluids that more or less flush out microbes, if you want to think of it that way. So, and again, this is that same unit I keep saying again and again. It's about the numbers. The same thing with cancer cells. We mutate cells, and periodically there are cancer cells for our immune system, and those wonderful killer T cells go along and latch onto them. It's why in healthcare we're dealing with sick people, and sick people frequently have what we call compromised hosts. And so those numbers are magnified because their immune systems are less effective. That's why I stopped taking immunosuppressant agents, because I felt that it's a catch-22. I'm willing to be sore every morning, good on some, worse. It goes with the territory. Just wait another 50, 60 years, you'll be old. Sure.
I feel alright.
You gotta have to. If you stop going to those bars at night, I know. It's like that. Don't start. My daughter got to start there. Remember, I told you the story about the fake ID. Great. Dad, how did you put up with me for all those years? I said, I have no idea how I survived. She should always apologize for all the trouble. Okay, here, Fimbriae, pili, that's how, for instance, the adhesion of, let's say, gonorrhea, different kinds of proteins. When you talk about slime layers or capsules, that's tuberculosis. Tuberculosis has that remarkably thick capsule that when you do the acid fast stain in lab, it's hard to do. And that capsule makes it very hard to find antibiotics for. It's very hard to penetrate. So that's why once it gets in to that macrophage, it makes it hard for it. Once the macrophage is able to swallow it, then big trouble. Then effectively, it's what I call hiding in plain sight. It lives inside the white blood cell. It flourishes, and eventually grows to vast numbers that burst and begin to travel all over the body. It's a bacteria that acts like cancer. So I'll frame it that way for you when we study in respiratory next. Okay. Viruses, protein spikes, worms. I don't do the worms, but when you see them, they have all those interesting things. A lot of people find those to be fascinating. Why? I don't know enough about it, but there's a lot of people do in this country, because it has a lot to do with sanitary conditions and water supplies. Okay? And the food we eat and ingest. Those people, we're lucky. You're not worrying about getting worms every time you go to the supermarket and get something to eat. The meat that you might get, the chicken you're not worried about that stuff. For years, the only product that people worried about significantly with parasites, and we don't see it today because they changed it, is pork. Pork had a disease called trichinosis. Okay, people were worried about that, and they went. A lot of genetic modifications were done over time. A lot of treatment was done, and it changed. Where pork became from something that was considered unsafe to being very safe. It's very yes, ma'am.
Is sushi ever a problem?
It depends on how it's sourced. Yeah, you don't. It's funny. I shouldn't say, because I'll go to Giant Eagle and grab sushi, but I get the sticks that are they're really fake fish. But if you're go to someplace that's responsible that does the sushi. It's not like backyard sushi. I'm trying to think of the right way to express it. You're dealing with raw fish, yes. It's what chefs call sushi grade. You're not using sushi grade. If you're doing absolute raw, and it's not sushi grade tuna or sushi grade salmon or whatever, you can be in big trouble. So we've gone to some high-end places that, I remember, because it was a birthday party, and went to some place in Station Square that was very high-end at the time, and had raw shrimp sushi and stuff like that. It was great. Nobody expensive, but I think I'd be very careful unless you knew what the source was. So when I do it, I just make like veggie rolls at home when I do sushi.
I won't do it.
No, I do it with just veggies.
Yeah.
I don't do it with raw proteins.
Okay, yeah.
If I
don't
put a
do it
protein in it, it'll be like a tofu-based protein. I know what's going on.
If the grocery store sushi freaks me out a little bit.
It does a little bit.
Yeah.
I'm sorry. That's why I mean those Californias, those spicy California rolls, not too bad. So much wasabi, so little time. I'm in it for the wasabi and the ginger and the pickled ginger. So I make them at home.
I'll eat the ginger straight up. It's so good.
Phagocytes, okay? Cells that engulf and destroy host pathogens by means of enzymes. We already knew about those. They have factors that fight them off. The more virulent ones can kill the phagocytes. The slime or the capsule makes it, and that's a big problem in respiratory, and that's really where tuberculosis. You're going to see things that cause pneumonia. The number one bacteria for pneumonia, Streptococcus pneumoniae. It's got a big capsule. Tuberculosis, big capsule. In the hospital, you deal with something called Klebsiella pneumonia, and it's a bug that's relatively tame in your gut, but it has an enormous capsule, and it's a very big problem in the hospital. And again, tuberculosis is an example. It's inside and multiplying inside a cell that's supposed to kill it. I mean, it's very clever for a single-celled organism. Okay, there are adaptations of a microbe uses to establish itself. And so here's how the ways that microorganisms cause damage. And this is, again, when we hear about direct and indirect, I was talking about spread. So when you get a question, okay, on a test, are we talking about how something spreads, like if the person next to you coughing and sneezing, direct or respiratory, or did it end up on a fomite? That's indirect. When you're talking about damage, direct is this, through the actions of enzymes or toxins, products, either endo or exotoxins that the agent produces. Okay, that's direct. Staff produces, and I talked about it in my lab, an enzyme called coagulase. It's nasty. It's what makes it so very nasty. It coagulates plasma, so the troops can't really get there to fight off the staphylococcus. That's direct action. It directly damages. It's like streptococcus producing a toxin that you get violently ill from. Okay, that's direct action. Whereas indirect, responding, it's your defenses, your immune system. And the biggest culprit for this, as you will see, happens to be the Streptococcus pyogenes. Most of the damage from that, your throat will be fine. It's not going to have any permanent damage from a strep throat. If it's not treated properly, you're looking at things like scarlet fever. You're looking at things like, we were talking about before, glomerulonephritis. You're talking about rheumatic fever. Very significant, potentially deadly complications. And they're mediated by your immune system. So direct is typically by toxins or direct damage from enzymes. And indirect is by your defenses, read that as immune system. Okay. And this is, when you talk about epigenetic changes, these are changing the markers. Once it invades that host cell, it changes its markers on the host cell. So it alters the ability of the immune system to attack it. Think of it that way. I mean, there's a lot of interesting phenomena that occur. And they're just showing you some examples of it. Secreting enzymes, either destroying the cells or weakening the boundaries. Exotoxins being produced. Okay, there's encapsulated bacteria, very, very difficult to deal with. Okay, can't be engulfed by the white blood cells, whereas that's their job. So there's a lot of these, and the epigenetic changes are when they're altering things on a genetic basis, so to speak. When we talk about enzymes, there are different ones. When we talk about exoenzymes, enzymes that are made and given off, secreted by the microbes that break down and inflict damage. They dissolve your barriers, lining cells, for instance, and promote the spread to other tissues. They are big players. Okay. Example, mucinase. Mucin is the protein that makes mucus. Mucus is a protector. What mucus has, when you all know it, it's sticky. And when it's sticky, it can trap particles. And then those particles, in the wonderful cilia cells, those pseudostratified ciliated columnar cells, travel it along so they can be expelled, swallowed, gotten rid of one way or the other. So if you take away the protective coating, now we have a problem. Hyaluronidase. Okay. You go back, and it may or may not have been emphasized when you looked at connective tissues lab. One of the things that makes connective tissues, creates that ground substance, is called hyaluronic acid. So effectively, if you can break that down, it can spread along tissue planes. So hyaluronidase, okay, helps hold. It's like glue, particularly in connective. This is one of the things that we sort of categorize magically as spreading factors. Going back to streptococcus, this is one of the things that's more of a direct mechanism rather than indirect by streptococcus. Streptococcus produces, it's interesting, if you have an infection with streptococcus, it's spreading factors. Start in one area and then begin to go, almost follow the circulation back to the heart. It's a very, very nasty kind of a process. And here's the other one I was telling you about. Pardon me. I forgot to bring a drink. With me this morning, coagulase. Nearly only found for our purposes in Staphylococcus aureus. So you did in lab. Did you do the coagulase test at all in lab as yet? I know my lab, we talked about, we didn't do it per se. When you're doing your unknowns in lab, and if you're one of your unknowns is Staphylococcus aureus, the way to prove it, more so than that MSA plate turning yellow, is to do what's called a coagulase test. We have it. All you do is you take some of the coagulase agents, which is rabbit plasma, and you pour a little bit of the rabbit plasma in a sterile test tube. I just pour off the liquid in a TSB, in a broth tube, because it's sterile inside. Put about a half a cc in there, half a milliliter in there, and inoculate it with some of your unknown, the gram-positive unknown. Incubate it overnight. It'll either turn gel-like or solid if it's Staph aureus, or it'll stay liquid if it's Staph epidermidis. And years ago, when I would get lab tests back from the labs, when I was in my office, this is 30 years ago, they'd come back, they would say, oh, that you have Staph coag positive or Staph coag negative. They wouldn't say aureus or epidermis. The big player was the coagulase. Okay, and kinases, again, another one of those spreading factors. Remember we talked about in anatomy and physiology, TPA. If you remember that, that's that tissue plasminogen activator, the clot buster. Before we made that in the laboratory genetically, what we did is we used streptokinase, another enzyme from streptococcus that was a spreading factor because it dissolved clots. So it was the clot buster we used to use before we had genetic engineering to make it. There's a whole lot of this stuff. Yes.
Okay, this might be a really stupid question, but does kinase make it like hemolytic? When you talk about hemolysis?
Yeah, no. No. The kinase, in general, when we use the term a kinase is an enzyme that activates things. When you hear the term phosphatase, it slows things down. Kinases generally send ATP to something and make it busier. I do not know why they call streptokinase. The only thing I can think of is that it must activate the clot-busting system. That's why they call it that. Hemolysis are different. Hemolytic enzymes are different. They, by definition, destroy red blood cells. And this is right back to streptococcus. So streptococcus and staphylococcus, the most common things that give us infections, are really a teaching lesson about that. Streptococci, and this is something I should have done in the lab when we looked at the plates. Dr. Dress talked about it, all alpha and beta hemolysis. Yeah. So hemolysis, enzymes that lead to that, you either do it partially, goblet, that's why we use the red blood cells. They can partially break them up. That's called alpha. Or totally. beta. No, it's not just streptococcus. Staphylococcus has hemolytic enzymes as well. But that's a tell. That's an indicator. If you have streptococcus that's beta hemolytic, that's where the strep throat strep is kind of most notoriously found. And we're going to look at that again in respiratory. It's here. Toxins. Oh, big time players. Okay. Specific, that is poisonous. That's what toxins are. Okay. And so, and there are all different kinds of toxins. Toxins that damage the kidney, nephrine. Toxins that we're talking about, alpha and beta hemolytic, are effectively toxins that destroy red blood cells. Enterotoxins, food poisoning. But food poisoning, the fancy name for food poisoning is food intoxication. We've ingested a toxin. It's not the same as a food infection, which put me in the intensive care. Even though I ingested that also. 20 years ago, I managed to get through. Neurotoxins, tetanus, botulism. Why they are so dangerous? They work very rapidly. Things spread a lot faster by way of your nervous system than they do in the bloodstream. The trouble with when they damage the nervous system is we have a word for that back in my old neighborhood, poignant. Poignant. When you grow up in New Jersey, you don't ask. Is that guy coming around anymore? No, he's gone poignant. Neurotoxins act on the nervous system. Hemolysis. I could have saved myself some trouble. You did this! Okay? Class of exotoxins that disrupt membrane red blood cells. They call them to hemolyze. This is complete. So this is, think of this as blood agar that you used in the lab. It's a good illustration. So this is, we call this is non-hemolytic. Fancy name, they might some authors will call this gamma. You know, it's like alpha, beta, gamma. Alpha is partially digested. Beta is complete. And like I said, if you saw at my lab, I'm holding up plates to the light. If you could see right through them, that's beta hemolysis. If it changes the color to a brownish or a greenish, it's alpha, and if it has no effect, we would call it gamma hemolytic. There are streptococci in all of those families. It doesn't, and for that matter, staphylococcus can do the same thing. So that's not an indicator. When you're trying to decide what bacteria it is, it's almost like I tend to call it a bacterial barcode. What we do is we make up what's basically called a dichotomous key. It's really a bunch of yes or no's. Is it gram positive or is it gram negative? Okay. Is it alpha hemolytic, beta hemolytic, or non-hemolytic? Okay. Is it an endoscore form? Okay. Is it a lactose fermenter? Is it coagulase positive? Is it catalase positive? All those yes or no's, and eventually you add up how many yeses and how many no's. It tells you which bacteria it is. That's kind of the way we do it. For years, there was something called Berkey's manual, capital BERGEY''S. Nowadays, it's in modern science, we have so many sophisticated tools that will tell us this. But when you're teaching the class or the lab, whether I'm doing it in lecture or doing it in lab, this is the way we used to do it. Okay. All the things I teach you about in lab, we did in our lab. And I had the best course I ever took was my micro class. The guy lectured worldwide on staphylococcus. It was really what we knew about. This is 50 years ago. And he was just brilliant. Give you an idea of how much micro we had. And maybe I told you, we had a micro lecture 4 days a week for like 4 hours each morning. And we had lab 2 afternoons a week for like 4 hours twice a week. The best way to do micro lab is 48 hours. You do the inoculations and you check them. The optimal time to check them is not a week later like we do, but 48 hours later. That's the optimal time to check them. That's when you get the best information. If not, really what we should do, it's just hard to do with the amount of students taking it. Take them all out of the lab and put them in the refrigerator and blah, blah, blah. Take them out to be at room temperature when you guys come to lab. But we don't have the resources to do all that stuff. It's very interesting. So I can remember that class so much, and unfortunately, I mean, it's the lecturer's son. He was a very close friend of mine. He was a resident who worked under me, and unfortunately died very young, probably in his 50s. Very sad. Endotoxin. You all learned about this. It's an endotoxin means it's not a toxin that's secreted. It means the structure, and that's that lipid A component we heard about the LPS layer, part of the outer membrane of the lipopolysaccharide layer. So go back and review that. This is systemic. It's systemic because it causes a severe immunological reaction. So even though it is a toxin, its effects are, it's an endotoxin, it's inherently part of it. Its effects are more or less indirect in nature because they work through your immune system. And your immune system absolutely goes nuts. Okay? And you go fever, inflammation, it can yield bleeding, it can yield diarrhea, all of these things. Blood infections by Salmonella, Shigella, Neisseria meningitidis, which is what causes bacterial meningitis, most common in adults. Salmonella, Shigella, and E. coli are all gut bacteria. The ones that cause infection are not normally found in your gut, or they would be very significant. They can lead to fatal endotoxic shock. That's what put me in intensive care. I had a salmonella infection that got into my bloodstream and caused a severe reaction. My kidneys shut down. I had a very substantial fever. Again, I'll just reiterate the story again. Severe enough that they called my 87-year-old mom and told her I was close. Scaring Ruth. That was wrong, Ruth. They shouldn't have done that. I'm stronger than that. So here we are. You can look through this. You can look through all of the things that does. Okay, stimulates antitoxins because eventually it'll form antibodies to toxins. That's how we fight those toxins. An endotoxin, it does not. Endotoxins are our immune system. Fever, yes. Okay, endotoxins are much more stable, meaning it's hard to make. You can see it's hard to make vaccines for them. As part of that cell wall, the liposaccharide layer, all gram-negative bacteria. And understand that most gram-negatives and most gram-positives that affect us, the way they do their damage is because of those exotoxins. I can't possibly read this if I tried, so I'm not going to try. Good Lord, my eyes are bad. Can you read this?
No.
Good. If there's something important, I'll make an effort to try to read that and see if there's anything I have to share with you. If not, I'm not going to get eye strain over it. And now, different kinds of infections. This is important. Okay. Localized are just that. They stay in a specific area. Systemic, they've spread. Typically by the bloodstream, but it could be through the nerves or even cerebrospinal fluid. So, for instance, most common infections, let's say you get a pustule. And the pustule is confined, maybe it turns into a boil where the pus is under the skin and it has to be lanced to be let out. Or it could be a wart, something relatively simple. Okay, that's a localized infection. Localized is good, okay, because generally they're fairly easy to treat. Systemic is not. Okay, and again, while the bloodstream predominates, okay, it can spread through it can spread by way of the lymphatic system, and certainly, as I was commenting on before, when infections reach the nervous system is a big problem. It spreads rapidly, and typically they are very destructive. So when we talk about things like tetanus or botulism or rabies, which I didn't mention before, those are all fatal unless they're treated promptly and appropriately. have to treat them aggressively with the antitoxin, or in the case of tetanus, you have to have appropriate tetanus, but you have to have the tetanus shot current, because that way, if it does somehow get into your system, your body's immune response will ramp up very, very quickly. Fight. And infectious agents is carried to it. So infectious agent spreads from a usually asymptomatic site and is carried to other. These are odd. Okay. Periodontal disease I don't know enough about it. I know my mom had it severely, had like 3 surgeries, and I did not know that the risk of spreading to cardiovascular sites. There's mixed infections are very problematic. We'll talk about them in wounds. Human bites, which I talk about in wound infections, are inevitably mixed. It's the old story. I would rather be better off with a dog bite or a cat bite. Dog bite being the least offensive, cat bites are a little more offensive. Human bites are very offensive. Human bite, even though they theoretically should have the same array of, they don't. Okay, they're very, very nasty. They always, always, always get infected, and typically it's a mixed infection and frequently anaerobic. They are a mess. Okay, primary is any, and secondary, and I talked about it a little, but I'll go over it again. The primary infection typically, when we hear about flu season, it's a viral infection. It causes a viral infection, influenza, that affects your lungs and your lower respiratory tract. The secondary infection is typically bacterial. In your respiratory system, primary infections are frequently viral and limited, where secondary infections, because of the damage that they do, are frequently very nasty. Okay, COVID was an oddball because it was a viral infection, though, even though it led to those, it was so severe immunologically because it was so new, being a novel virus, that it could be fatal in and of itself. And then acute comes on quick, goes away fast. That's chronic. That's acute rather. Chronic, basically when it's quite progressive and persists over long periods of time. And we'll begin to look at that. I'll leave it here. I have some other slides I'll go to about acute and chronic. Have a great weekend. Bye-bye, you two out in virtual land.